Thank you, Jason. Hello, this is your moderator, Julian Heath again. So we have a few minutes left in this webinar for our Q&A. So if you'd like to ask Jason something, please submit your question now. So Jason, are you ready for a few questions?
Yes, Julian, thank you.
Okay, now the first one. With chip scanner, how does the depth profiling work? In other words, how do you get to the next layer physically once the information on the previous layer has been obtained?
Okay, the chip scanner itself is a scanning electron microscope only. It is not a deprocessing instrument. So the depth profiling, each layer is deprocessed layer by layer using other equipment, including chemical mechanical polishers, ion etching, and in the case of collecting the images of neuronal tissue, some ultramicrotomes are separately used.
Okay, thank you. The next question, how do you compensate for drift in the scanning EM?
Okay, drift. Yes, when you are collecting images over long time frames, any scanning electron microscope will drift, and the chip scanner is a scanning electron microscope, and it's impossible to build a microscope that doesn't drift. So this is a common problem for electron beam lithography as well, and so we really understand the problem well. And so the chip scanner 152 has an environmental enclosure surrounding the entire instrument, and that maintains a temperature, a very tight temperature tolerance inside the enclosure where the vacuum chamber is, where the electron optics are, and even the scanning electronics. All of this has very tight temperature control, and that really minimizes the room temperature variance that is coming from the environment. Furthermore, there are automated drift compensation routines that are standard for e-beam lithography instruments. And these drift compensation routines are readily available for the electron for chip scanning, for capturing these images over large time, over long times. So if the images are being captured, and if each image takes some time, and you can set the software to go and correct for any drift, let's say after every 10th image or after every 10 minutes. So it's quite variable. Also depending on the sample. The sample can also cause drift by charging as well. So there are several tools available to compensate for drift, and then we try to build also a stable system. Finally, the laser interferometer stage is also a very stable stage to begin with. So that, coupled with temperature control, makes it an inherently stable instrument. So.
Yep. Okay, nice answer. Next question. With the high precision stage and a calibrated image, what sort of overlap requirement would you have to consider?
Okay, so the in a few slides prior, we showed quite a large overlap of about 1 micron, but that's completely overkill, if you will. A typical overlap that we start with is, let's say, 100 nanometers, and if we need any more, any less overlap, it depends on the overall imaging conditions. So if there's a lot of sample charging, or if it's, you know, if there's any other source of drift that is giving us problems, you might need some more overlap. If there if the images each take a very long time to capture, like if you're capturing a 50,000 by 50,000 pixel image, that would take a long time, and so you might want additional overlap there, unless you're correcting for drift after each image.
And here's a sort of supplementary question. How long would it take to image and stitch 22 nanometer sample images? Obviously, that's a kind of an open-ended. But can you give us an example of a typical run?
Yeah, so it is open-ended, and it really depends on the overall area that you're trying to cover. I don't have specific numbers for the images, or even the number of images that were captured for that chip. However, the instrument is capable of automatically capturing these images, and so it can run for days in quite a robust way, just continuously capturing these images. But it really depends on the acceptable signal to noise for the application, and a variety of other settings to determine exactly how long it takes to capture each image.
Yep. Okay, thanks, Jason. Next question. What type of SEM was used in the work that you described in your talk, and also what is your definition of high resolution SEM?
Okay, so the SEM is thermal field emission SEM, and the beam diameters are comparable to traditional thermal field emission scanning electron microscopes. So I would say of the order 2 nanometers is like the beam diameters that we're talking about here. So it's definitely high resolution field emission scanning electron microscopy. And in addition, there are a variety of voltages very similar to a traditional scanning electron microscope, also with some of the inherent features that you would expect in terms of doing different scan rate.
So, for example, in the brain mapping project, you're clearly able to resolve the plasma membrane of each neuron, because that's in the order of 7 nanometers or so.
Yes, that's correct. The myelin sheets and the small features, these can be resolved. Yeah.
Another question. So we talked about drift. How do you maintain focus during these long operations?
Okay, yes. So the larger area that you scan over, the more the sample can get out of focus. Now, we have 3 different ways of controlling the focus over large areas. One is using the laser height sensing, where the laser beam is bounced off the sample, and its reflection is recorded, and the position is determined there. And that measurement can be done well within the depth of field of the scanning electron microscope. It's also possible to use a simple planar correction, and finally, it's also possible for the chip scanner 152. In situ, it has the ability to level the sample by basically moving up and down the 3 points on which the sample holder is resting.
Okay, one last question. What is the maximum field size that you can acquire? Again, a bit of an open question, I guess.
Yes. This is, I think, our current limit for the chip scanner. It's a 16-bit machine, and so 16 bits corresponds to about 50,000 pixels. And that's why the instrument has the limit of capturing 50,000 pixels by 50,000 pixel images. Now, if you take a 2 nanometer beam diameter and you multiply it by 50,000 pixels by 50,000 pixels, you end up with a 100 micrometer field of view. Now, it is possible to go to larger fields of view if you increase the step size or the size of the pixel. But if you're matching the pixel size to the beam size, then 100 micrometers.
Okay, thank you, Jason. Well, unfortunately, we've now run out of time, so that ends our Q&A. So this time, I'd like to thank everyone for attending today. Also, special thanks to today's speaker, Jason Sanabia, for his excellent presentation. I'd just like to remind you all that today's event has been recorded and will be available for viewing on demand in a few days' time. There will also be a feedback area should you have any further questions or comments you wish to make. An email with this information will be sent to all of today's attendees. The Microscopy and Analysis team are planning more webinars, and we hope that you'll be able to join us again for our next presentation. So that concludes today's event. Many thanks once again for your attendance, and enjoy the rest of your day. Goodbye.